Hello, good evening, everyone, and welcome to Café Scientifique. My name is Jennifer Cobb. I'm the host of the speaker tonight, Dr. David Goodlett, who will be talking about what doesn't kill us makes us stronger. I am a professor in microbiology and biochemistry in the Faculty of Medicine, or in the Faculty of Science at the University of Victoria, and the Faculty of Science is supporting this event. So I will give a few words on Dr. Goodlett, who is a colleague. And when I was reading about what he's done, you know, with inside work and out, he's hopscotched back and forth across North America, both in life and in his professional career. His professional career is illustrious. And I also learned he's a semi-professional musician, so maybe after this performance he can give us another one. And without further ado, I will hand it over to you.
Okay, you heated me up. Okay, good evening. Thanks, Jen. And what to say? So I'm supposed to talk about, well, you have to give a title and an abstract, right? And we don't normally talk to lay audiences, so I am going to talk about sort of what doesn't kill you makes you stronger. But on the way and around doing that, what I want to talk about is what our lab does, and so this will set you up for the end, because I'm going to tell a whole bunch of stories that may seem like they're not connected. And they are in my mind, but they might not be in your minds. But so I'm a chemist, and I use mass spectrometers to study the structure of molecules, and specifically molecules that we find on the surface of bacteria, lipid molecules. And lipids are, fat soluble, like butter, olive oil, that class of molecule, right? Not water soluble. So we use mass specs to study these molecules, and these molecules happen to actually be recognized by our immune system. So they're immunostimulatory, right? But before I get onto that, I just want to tell a couple of origin stories. So Jen already let the cat out of the bag about my failed music career. But so I grew up in Montgomery, Alabama, and you would pretty soon recognize I'm not from here, right? I have my very American t-shirt on. But I grew up in Montgomery, Alabama, went to a public high school, and my chemistry in high school sucked. It was not good. It was like elementary school chemistry. And but I played music all through high school, and I thought I was going to become a musician. And I got to university, and all of my buddies were going to be doctors, lawyers, pharmacists, optometrists, dentists. And my guidance school teacher had told me that, oh, you should just learn a trade because you're not going to amount to anything. I'm serious. And it was because I was a B student, right? I was not interested in most things that they were putting in front of me. And I got to university, and I started music in university, but I took, this is weird, but I took chemistry as an elective. Yeah. So already, I'm not normal, right? He's an American, he studied chemistry as an elective. So, and it was like, oh, a light bulb just went off. I mean, I almost failed the first term because it was so hard and rigorous, and I just didn't have any experience with that from my public school training. And, but I just was mesmerized by the fact that, molecules recognize each other, and they either react with each other and make something new, or there might be an explosion, or they don't, right? And you can have these molecules swimming around together. Some of them recognize each other, and something new happens. And in other cases, they just completely ignore each other. And I just could not get over that, right? So I was just smitten right from the start with this concept of molecular recognition. Why do certain molecules recognize each other? Like I still recognize my wife back here, and that's actually chemistry, right? There's chemistry going on in my brain that allows me to recognize her for now. Yeah. Yeah, so that's chemistry, and it's completely geeky. I had no idea what I was getting into. I didn't know that you could grow up to be a professor, but I did. I just sort of fell into it because I was curious, right? Curiosity killed the cat, and that's basically me. I can't not ask. It's a double negative. I can't not ask questions, and so as I'll get onto a little bit later, our lab is studying the structure-activity relationship of these molecules that we pull out of bacteria. And some of those molecules stimulate your immune system to make you really sick. Like if anybody's ever encountered salmonella, I'm sorry. But you would have gotten really sick, throwing up, vomiting, diarrhea. And part of that's an inflammatory response that your immune system triggers to try to kill the invader, so to speak. But so if we back way up, before I went to university and studied chemistry, my parents didn't use a condom, and that's that's actually why I'm here. And when we give professional lectures, we're told not to tell jokes like that. So I might, overdo it tonight. But the thing that's directly related to, sex is microbiology, if you don't know. And public service announcement number one. I'm not a physician, so I'm not. This is my opinion, right? Not my. There is a doctor in the house back there someplace. So if you need some real medical advice, I can point you in the right direction. But yeah, sex is microbiology. And over the course of, the last decade, it's become almost impossible to catch HIV. But the oldies and the goodies are still around, gonorrhea and syphilis. You can still catch those. And so this is partly why our lab is studying infectious disease. But back to my story. So it turns out that before I was 4 years old, I had pneumonia 3 times. I was in the hospital over and over again, and antibiotics saved me. And to listen to my mother tell the story, it sounds like what was happening was I was having this allergic reaction and just massive sinus drainage, right? Just drowning in sinus drainage. I'm in Alabama, and there's lots of pollen, of course, down there, more than here in the springtime. And of course, your lungs are really the first line of defense against the bacteria that you encounter every day, right? You're inhaling things constantly. The bacteria are everywhere. You can't get rid of them. And you're inhaling in them, and your immune system basically takes care of that, right? What's amazing is we mostly don't get infections, right? Because you're just killing the things that come in. But if your immune system gets overwhelmed, like it did when I was a baby, because you're swallowing all this stuff, your sinuses are just full of bacteria and viruses, and you're swallowing it, but you're aspirating some of it as well, right? So some of it ends up in your lungs. And if you're a baby, you can't communicate, so eventually you're just hacking up, green stuff, and you're turning blue, your mother's freaking out, and you go to the emergency room. And I was born in 1960, so, you got antibiotics, thankfully. Eventually, they figured out that antihistamines would basically prevent me from having this problem, right? So I spent most of my young years jacked up on Benadryl, on into my teens, right? Every spring, if I didn't start taking Benadryl, say in February, March, I would have a chest infection, right? So, and that's just, normal bacterial flora that's all around us. So this got me along with the chemistry that I eventually realized I was quite interested in, I just was lucky enough to meet some microbiologists, and as an analytical chemist, so that's how I see myself anyway, someone who solves other people's problems, basically. I'm sort of like the engineer on the Formula One team. I'm not driving the car, I'm not designing the car, but I'm figuring out what's wrong with the car and trying to solve problems. And so I started working with some microbiologists to solve problems that they had that were related to these lipid molecules that are on the surface of gram-negative bacteria. And I'm going to have to get my REI. You guys have Mountain, what is it? Mountain Equipment Co-op? Yeah. MEC, yeah. This is REI. It's just tea. Yeah. Yeah, so I ended up with sort of this interest in molecular recognition, and then I realized these microbiologists had this really cool set of problems, that these lipid molecules would activate your immune system or not activate your immune system depending on their structure. And so I'm supposed to talk about chemistry without really talking about chemistry, but if I describe these molecules, I can try to do it with my hand. So everybody knows what glucose, sucrose is, right? So these lipids, there's a lipid called lipid A that's in gram-negative bacteria, and that lipid A is recognized by something called a toll receptor. And so there's about a dozen toll receptors. It's spelled just like toll for the road, right? And it's basically your innate immune system has a bunch of these toll receptors that are programmed, right, in your DNA to recognize foreign invaders. And one set of those foreign invaders are gram-negative bacteria, and there's this lipid that's in the exterior, the outer membrane of those bacteria called lipid A. It's named very cleverly because chemists, they, yeah, so you must know some chemists. So anyway, when they were in the old days, when they would try to understand why a molecule had some activity, they knew that the bacteria had activity. They knew it was related to some molecule, so they would fraction it by chromatography. Hopefully, you know what chromatography is. But anyway, they would produce these. They would separate these molecules that were all very similar, right? And the one that had the most activity was the one in fraction A. So they literally called this really important molecule lipid A. And so lipid A is responsible for activating toll receptor number 4. And it sort of looks like this. If my thumb and my pinky are negatively charged, like they're phosphates, tell me if I'm getting too far into the chemistry. And then the surface of my hand here is a sugar, right? And then my fingers here in the middle are fatty acids. So the lipid part is my fingers here. And so this sits down in the lipid membrane of the bacteria. When the bacteria get inside us, there are various mechanisms to transport that to the toll receptor. And so the toll receptor has this cleft. The toll receptor is a protein. It's got a cleft. And the lipids sit down there in it, and it's kind of like Goldilocks' pudding. It's either really hot, too hot, or not hot enough, or just right. And if it's just right, like E. coli or salmonella, you're going to get sick. If it's not very hot, I'll talk about this a little bit later. There's something called Yersinia pestis that caused the Black Plague. It doesn't activate right away, but eventually it activates. So this molecule is rather simple molecule, some phosphates, right? Coca-Cola's got a lot of phosphoric acid in it, so these are just literally phosphates like phosphoric acid, some sugars on the backbone, and then fatty acids. So when these bacteria encounter different environments, they can completely remodel the structure of this molecule. And they can make it either agonistic or antagonistic, so it either activates or it doesn't activate this toll receptor. And the toll receptor is responsible for initiating inflammation. So you probably heard about inflammation in the news in various ways, right? Everybody's, well, I think in terms of the general press, you would probably hear about inflammation in terms of keeping it down so that you live longer, right? Inflammation is associated with aging and maybe cancer and other things. So some of these molecules will immediately stimulate a really strong response, like salmonella, if you get food poisoning, E. coli, like if you hear about people dying from a spinach outbreak, it's because there's some cow poop on the spinach, and that cow poop's got a lot of E. coli in it, and there's certain strains of E. coli that are really quite dangerous. And it's generally true that the older you are, the worse your immune system is at dealing with these insults, right? So part of that response, that inflammatory response, is related to these molecules at our lab studies. And the structures can change really quite slightly, and the activity changes quite a lot. So what to say, these molecules basically stimulate, in the case of toll receptor number 4 and some of the other toll receptors, cytokines. So you might have heard about cytokines. There's various kinds. They're really small proteins that cause inflammation, and inflammation is targeted at killing cells, right? So that the inflammatory response that your immune system starts is geared at trying to clear the invader, the pathogen, right? Your immune system recognizes that it's bad, it shouldn't be there, and inflammation starts, and some of the cells are killed, hopefully most of them if it's a pathogen that, that's easily recognized. So TNF-alpha is a cytokine, and it's one of the predominant cytokines that's produced in people that have Crohn's disease. So people with Crohn's disease can go into remission by being given an antibody, right? So I'm sure you've heard about antibodies. We make antibodies to foreign invaders, right? That's what a lot of vaccines do, is they give you an antigen that's the target, and then your body makes an antibody against it. In this case, people figured out how to make antibodies and make them into drugs, right? And so Humira is one of those antibodies that binds up TNF-alpha. I think it's the biggest selling drug on the planet, right? It's billions of dollars that they make. And in fact, it puts people with Crohn's into complete remission for a while. The problem is it's foreign, so eventually your body starts to make antibodies against the antibody that you're being given. So it doesn't work forever. But it works. And so I've gotten a little ahead of myself because I'm not reading my notes because I don't memorize anything. This is why I was a B student in high school and nearly failed chemistry. I've got to figure everything out right from first principles. So people with Crohn's, right, their gut, all of our guts are lined with immune cells, right? And normally everything's fine, it's in check. You've got some flora and bacteria in your gut, and this immune response doesn't happen the way it happens in Crohn's patients. But in Crohn's patients, they develop these little perforations of the gut that cause them to have fevers and other sort of maladies that can be quite serious and dangerous if they're not dealt with. Another way to put those people into complete remission is to give them a stool transplant, which sounds disgusting. But in fact, if you give them a stool transplant with somebody who's got a healthy gut microflora, that will also put them into remission, but only for about a month, maybe 6 weeks. Oh, yeah, sorry.
Oh, no, I'm just meaning general.
Yes. So these people with Crohn's actually have options now that they didn't have, a decade ago. But that inflammatory response that causes perforations, that causes those people, to not feel well in many ways, is related to inflammation, right? So I'm supposed to talk about what doesn't kill you makes you stronger, And we should talk about the word infection, right? Because the word infection is, it's a bit weird. I mean, we're completely infected all the time, right? There's more bacterial cells in us than there are human cells. And physicians only consider it an infection when something's gone wrong, right? It's when one bacteria has outcompeted, or yeast or virus has outcompeted everything else and is causing a problem, right? Like when I was a baby and I had these infections, right? That's obvious that there's a problem, right? It's not normal. The homeostasis is basically broken, but normally the bacteria all get along. At some point, when you've got an infection, right, one bacteria has outcompeted everything else for the resources that are available. And so to go back to the Lipid A story, I want to tell two stories. One has to do with cystic fibrosis. And so people who have cystic fibrosis have a defect in a chloride transporter gene. And and it's basically a disease of Northern Europeans. So probably, some of you may know someone who has cystic fibrosis. There's a chloride transporter defect, right? And what that inevitably what that means is that inside the patient's lungs, there's a change in ionic strength. And because this chloride has to do with magnesium and other just electrolytes that need to be in a certain ratio in the lungs for the person to be healthy. And so in those people, it's not quite right. And what happens is, even though all of us, every day when we're in the shower, we inhale something called Pseudomonas. And Pseudomonas is not the pink stuff that's growing in some of your showers. That's a different bacteria. It's not dangerous to us either unless you're immunocompromised, but most things are a problem if you're immunocompromised. So Pseudomonas is everywhere. It loves the water. We inhale it. Macrophages, our immune system, just slay it, right? We're constantly inhaling it, and we kill it. But if you've got cystic fibrosis, the organism now, amazingly, it's just sensing a change in the chemical environment in the lung, but it remodels its membrane. And when it remodels its membrane, it changes the structure of that lipid A molecule that's in there. And as a result, this doesn't trigger that killing response as well as it should. And so those bacteria now grow in biofilms that they don't grow inside of most of us, right? And so it basically, it isn't in and of itself responsible for those patients dying, but it does cause quite high morbidity, right? Because they're unable to clear their lungs, at least, well, so I'm 63. When I was in the 60s, you would see on TV people pounding on the backs of kids that had cystic fibrosis to try to clear all that mucus. And so that response, that buildup of phlegm and mucus, biofilms, bacteria, is partly related to this molecule lipid A that our lab studies. And so the interesting thing as a chemist is this gets at host susceptibility, right? So I forgot to say. So like if you get an infection, then it's like an algebraic equation. So there's virulence of the organism, how strong is it? There's susceptibility of the host, and there's the number of bacteria or viruses that infect you. And the number or the load that you get, that's actually the easiest thing to measure, because they can get healthy, normal volunteers to agree to be dosed with various amounts of things like COVID. And that's how we know that, like COVID, which is a virus, not a bacteria, you only need about a few hundred particles to become infected if you're a normal, healthy person. If it's norovirus, which is a bad disease, you only need 10, 12 particles. Right? So every organism has a different sort of virulence to it, or the number of molecules that are required to create an infection. So one of the things that's difficult, though, to understand is host susceptibility, But there are a few cases like cystic fibrosis where we know, we can explain now what's going on, right? And so it turns out if you take that Pseudomonas out of the lungs of the patient and you put it in one of our mass spectrometers, we can see what the lipids look like. We see that they have a specific structure. But then if we take the same Pseudomonas and we grow it out on a Petri dish, it reverts. It begins to look exactly like the Pseudomonas that grows in our showers. So the Pseudomonas normally is not harmful, and it's only harmful to those patients because inside them they have this situation that allows it to change, and it's just trying to survive, right? But in surviving, it increases the morbidity for those patients. And so we can see an immediate change in the structure of that lipid A molecule from looking at it directly from the patient to looking at it after it's grown in the lab. Yeah, somebody said, wow, that's like me too. That's like my every day. I wake up and I'm like, wow, unbelievable. Yeah, welcome to my life, yes. So I should say I have another story to tell about a different bacterium, but I want to explain a little bit about the tools that we use. So, and the tool is called a mass spectrometer. Has anybody heard of mass spectrometers? Probably, yeah. So I'm going to insult you people, but the rest of the folks I'm going to try to explain. So mass spectrometers allow us to measure the mass, or we say molecular weight, of things that we can't see. And these lipids that we work on are too small to see. Like with microscopes, you can see organisms. With electron microscopes, you can see molecules. You can see some proteins. So we're actually quite good now at looking at really big molecules. But these lipid molecules still quite difficult. So we use mass spectrometers, and there's a bunch of different ways, different kinds of mass spectrometers. I mean, humans are clever beyond belief, right? Every time somebody files a patent about a way to do something, somebody else comes along and files a new patent that says, no, I can do it better, and I don't have to obey your patent. So one of the ways we can use to measure mass is frequency, right? So everybody's seen someone jump off a diving board, right? Probably. And so if you think about what happens, right, the diving board oscillates up and down, right? So if you took me and you dropped me 10 feet above the diving board, then the diving board would oscillate with some frequency proportional roughly to my mass. Remember, I'm from Alabama. We use a lot of duct tape. So if you duct taped 2 or 3 of me together, then I would be 2 or 3 times bigger. I would weigh 2 or 3 times more. And now if you drop me from the same height, right, the board's going to oscillate at a higher rate, right? So we can calibrate these kinds of frequency responses using molecules that we already know from other ways, what they weigh. So we use mass spectrometers to figure out what the weight of the molecule is, and we also figure out, and that tells us something about it, but then we also need to, it's like knowing that you're all humans. I assume you're all humans, but it's not much more than like knowing you're all humans. In order to know what kind of human, right, then we need to sequence your DNA. In our case, we need to figure out the structure of the molecule. And so we do that literally by just blowing it up into bits. And then we can put it back together, but we can only put it back together if we understand the rules, and the rules are related to the kind of molecule. So it's kind of like if you're an archaeologist and you came up on a building site and there's nothing but ruins, right, just blocks of stone, and you look closely, maybe you see that there's no arches, right? There's no arches anywhere in the ruins. And right away, okay, there's no arches. So this probably is before the Romans, because the Romans figured out how to make really strong arches. Like the Mycenaeans only had these post and lintel-like archways, right? Not archways, but entryways. So if you're an archaeologist, you can reconstruct what the building probably looked like by knowing something about, who you think built it. So we do the same thing, right? We have a library in our minds of what we think these molecules, what we know these molecules look like, and when we encounter an unknown, we blow it up into parts, and it's kind of like a jigsaw puzzle, or if you built something out of Legos and it got knocked down, then you've got to put it back together, and that's basically what we do. So, and we do that literally all day long, almost every day. And we're constantly looking for new organisms, new structures, because every structure has a function. And I should say that in the lab, we can actually measure function, right? So I just told you how we assess structure by using these mass spectrometers, like to figure out what the jigsaw of parts looked like before we blew it apart. With function, we basically create a cell line that has got this toll receptor in it, and it's got a reporter that probably could just be like a blue color. And when you expose that cell line to some of these molecules, then the amount of blue color is related to the amount of cytokines that are produced, right? So as chemists, this is amazing, because now we can not only just figure out the structure, but we can associate that structure with a function, right? And we can quantify it, right? So we do that. We've measured, we're working on a paper where we isolated a organism out of shark poop, and we've characterized the lipid A structure. This particular lipid actually doesn't activate toll receptor very much. We don't know exactly why. We have some clues. But so the point is, we spend a lot of time looking in new places for organisms to try to uncover new structures. And the reason we'd like to uncover new structures gets at therapeutics. Like we'd really like to understand the relationship between the structure and the function more closely than we do. Like we have a broad idea of what's going on, but we're not so good as like the antihistamine story, right? So with antihistamines, it looks a lot like histamine, and it sits in the receptor, and now histamine comes along when you have an allergic reaction, and it can't go into the receptor, right? The antihistamine is sitting there, and it's not going to come out because it's bound tightly, and it prevents the histamine from producing that reaction. And so when I finally get done in a few minutes here, I will have gotten through a story about sepsis, and what we'd really like is to use these lipid molecules as an antiseptic therapeutic. So one last story that is frightening, and that has to do with the Black Plague. And so the Black Plague in Europe happened around 1300 or so. And it was nothing like COVID. It was, 100 times worse, right? Tens of millions of people died from the Black Plague. And so the interesting thing as a chemist is this Yersinia pestis is the bacteria that caused the Black Plague. And it still exists in the world, like Madagascar. It's endemic in Madagascar. But in the modern world, the Yersinia pestis is kind of a wimpy molecule. If you know you've become exposed, you just get antibiotics and you're going to be fine. But of course, in those days, they had no idea what was going on. People were just dying because it was spread by various means. But it was like COVID; it could be spread through the air. So these lipid A molecules that I've been talking about, they are in Yersinia, and in Yersinia, it's got two different worlds it lives in. It lives in insects, and it lives in mammals. And the difference in temperature is about 10 degrees Celsius. So inside an insect, this lipid A molecule in Yersinia becomes a structure that our host immune systems would recognize and kill. But inside of us, that doesn't happen. The Yersinia, because now it's 10 degrees higher, it remodels its membrane, and it makes a lipid A structure that isn't recognized by the immune system. And so what does that mean? Well, it means that the Yersinia grows out of control, right? Just billions and billions of Yersinia grow over the course of a week. So if you're infected on Monday, you're not going to feel any different until maybe Thursday or Friday. But if you don't go to the doctor Thursday or Friday, you might not survive until the next Thursday or Friday, right? So the problem is that there's a structure-function relationship with these molecules, but there's also a concentration-dependent relationship. So at first, the lipid A from Yersinia sits in the toll receptor and just doesn't do anything. Nothing much happens. But by the end of the week, there's so much Yersinia present that it begins actually to start to trigger an immune response, and you get a fever, you start to not feel well, right? And the problem here is, has anybody heard of the cytokine storm? You probably heard about that maybe during COVID. So the problem is these people can go, COVID patients or Yersinia infected patients can develop sepsis. So, sepsis is just a bloodstream infection. So if you had a gunshot wound, so before I was here, I was in Baltimore. There's 300 murders a year in Baltimore. And so I feel quite safe here. I have no adrenaline rush here. It's just like very strange. But the British military send their surgeons to Baltimore, to the hospitals to train. There's so many gunshot wounds. It's true. Yeah. But if you get a gunshot wound, then all of a sudden, you've got your bacteria is loaded with bacteria. Those bacteria now can get where they're not supposed to be, which is outside this immune protected zone, right? And so you could develop sepsis. So what we'd like to do is develop one of these lipid A molecules. It's kind of like an antihistamine, but antiseptic. So it sits in the toll receptor, and it prevents the molecules that are from the live bacteria that are in you from triggering the response. And so why did I mention cytokine storm? Well, most people who have a problem with sepsis, they're not dying from a bacterial infection. So it turns out that, again, we'll start on Monday. If on Monday the physician thinks that you have sepsis and they give you antibiotics, you've got like a 90% chance of surviving, right? The antibiotics kill most of these bacteria in these patients, not a problem. But if you wait until Friday, now just think about the Yersinia story, the bacteria have continued to grow, right? So there's billions more bacteria inside you. And so by Friday, 5 days later, your chance of survival is only about 10%. And this is not, you're not going to die from the bacterial infection. You're going to die from this thing called a cytokine storm. And so what happens is the physician gives you antibiotics that kills the bacteria, but your body can't get rid of the cellular debris, right? So all these lipid molecules, it's not just lipid A. Remember, there's 12 something different toll receptors, and they all recognize different molecules in bacteria and viruses and fungi. So all of those things now are there, and they can't be cleared fast enough, and they just turn your inflammatory cascade on, and it can't be turned off, right? And so this is what kills most people who suffer from septic shock, not the bacteria directly. So this is why we're trying to understand the relationship of the structure to the function of this molecule so that we could create a designer molecule that sits in the cleft of toll receptor 4, and your physician could treat you with that first. Like if you've got a gunshot wound, well, you just get this antiseptic molecule, and they're going to give you antibiotics, stitch you up, give you antibiotics, and you're not going to die from the cellular debris that's going to cause this massive cytokine storm. What else was I supposed to say? Let's see. Cytokine storms, that's almost the end of my notes here. And I'm sure I left something out. Oh, yes, so what does our lab do beside the structure function stuff? Well, we actually have a pilot study at Vancouver Island Health Authority to use these same lipid molecules as a diagnostic, right? So I just described how to use them as a therapeutic to prevent cytokine storms, but it turns out that we can use them as a diagnostic because mammals don't make these molecules. If you're suspected of having a urinary tract infection, and you go to the doctor, you pee in the cup. Normally what happens is the analytical lab takes that urine, they plate it out on a petri dish. That takes about 12 to 24, maybe even 48 hours before something grows. They pick a colony, they actually put it in a mass spectrometer, and that mass spec reads out protein patterns, and it tells you what the organism is. But that takes several days, right? And if they suspect you've got sepsis, it actually takes an extra day because the physician is going to take some of your blood and put it in what's called a blood bottle. And inside the blood bottle, there's just nutrients. It's just a tiny little bottle, you know, like yay big. And it'll grow for about 24 hours, the bacteria that are in there. And then they take that out, they plate it on a petri dish, and they grow it for another couple of days before they can tell you what it is. And so what our lab, we're not testing anything to do with blood bottles yet, although I hope, Bruce, that we can convince Michael, my colleague at VIHA, to do that. But right now, we're testing it on suspected UTIs. And so it turns out that, at least in British Columbia, that almost all suspected urines suspected of being urinary tract infections will go to the analytical lab and be tested. But something like 70% of them are negative, amazingly, right? People feel something weird, the doctor suspects something, but there's no infection found. And that still takes several days, right? So with our lipid-based mass spec assay, we can, as soon as you've peed in the cup, we can take some of that, put it in the mass spec, and tell you exactly what's going on. Yes, you've got an infection. No, you don't have an infection.
One day.
Well, within an hour, yeah. So, but the point, well, yeah, there's always politics involved, and there's big money involved, and look at me, I don't have big money. But, you know, seriously, yeah, though people have skin in the game, right? And so there's a lot of money that's gone into the way that things are diagnosed now. So there's always resistance to change. Anyway, we're still testing it. We've got some things to work out with it. But on first blush, it appears to actually work quite well. What else? Now, all this interactive stuff got me off course. That's okay. Yeah, so UTIs. Oh, I know the other thing that shocked me when I started working on this problem was something like 20% of UTIs go on to become sepsis. It's amazing, right? So it's actually really a serious problem that demands diagnosis as soon as possible. Okay, I think the only other thing that I wanted to say was our labs also, we're looking for microbial signatures all over the place, and we're basically, like in that Pseudomonas cystic fibrosis problem, we're trying to develop analytical methods that allow us, you know, to take whatever's on this microphone here that I've been breathing onto, which has now got bacteria on it that have come out of me, and take those to the lab and tell you what's there, right? We're trying to avoid culturing in the lab because we know from the cystic fibrosis Pseudomonas story that this completely changes the molecules that we're going to look at, and it completely changes their functional sort of categorization. The other thing that I think is interesting and terrifying is that, I mean, literally bacteria are everywhere. And now I promised my wife I wouldn't talk about vaginas tonight, but whoops, sorry, honey. I'm almost done. So a normal healthy vagina has a low pH, and that's actually maintained by a bacteria called lactobacillus. And so bacteria are everywhere, right? And we don't know nearly as much about what's going on in men's prostates, partly because it's quite a bit more difficult to get to than a vagina. But anyway, what's frightening is that people have found oral pathogens in that, like Porphyromonas gingivalis, everywhere, right? So the Porphyromonas gingivalis, they don't know that we describe them as an oral pathogen. They're just bacteria, and they're inside us, and they can go wherever they want. And so there are oral pathogens that have been found in prostate. There are all kinds of bacteria that have been found in ovaries. And so I think, and it's known that some bacteria cause cancer, like H. pylori contributes to some cancers, GI tract cancers. So I think what's going to happen in the next 10, 20 years is I'm going to retire, honey, I promise, before 10 years is up. But what you're going to find is that this connection between inflammation and cancer and chronic inflammation, I mean, right, that's just, it's not killing you immediately, but it's contributing to changes in your immune system and your cells over time. We're going to understand a whole lot more about that in 20 years, maybe even sooner. I mean, there are really cool papers that are being published right now that have to do with inflammation and cancers, and it's really quite exciting. And we're working with people at BC Cancer on ovarian cancer, which is, unlike prostate cancer, is actually a seriously unmet challenge. If you look back over the last 50 years or so, 90% of the men who get diagnosed with prostate cancer, they survive 5, 10, 20 years. But it's not true with women and ovarian cancer. And I'm not saying that there's any connection between microbes in either one of these cancers, but we do see, when we look in certain ways, we can see that there are microbes present in these tissues. And we know that microbes cause inflammation. And so I suspect that over the course of the end of my career, there are going to be some exciting discoveries that take place on this axis of inflammation and microbes and chronic inflammation and cancer development. Okay, I think that's all I want to say. So I'll bring Jen back up. She's supposed to moderate here.
Okay. Thanks very much, Dave. I learned a lot tonight. Questions? Yes.
When you blow it up, how do you know you've got it right when you put it back together again?
So I'm going to repeat the question for the people online. So the question was, when you blow it up, how do you know?
How do you know when you put it back together again that it's right?
How do you know when you put it back together if it's right?
Yeah, good question. So we have a library of, I don't know, a few hundred molecules where we know what the jigsaw pattern looks like when it's not together and when it is together. And so the truth is, we don't really know. We sort of know, and we just make educated guesses. And then we couple that analytical work with the function work and the cellular assay. And if we see changes that we know from prior structures should be pro-inflammatory or anti-inflammatory, then that makes sense. And if we see changes that don't make sense, then we'll say, this is probably not correct. Or sometimes, it's rare that we deal with a single molecule. So, nature isn't like Western medicine. You take an aspirin and it has one effect, or you take an antihistamine and it has one effect. Nature presents an array of similar molecules to receptors, right? So it's often the case that we're not just blowing up one molecule, we're blowing up 4 or 5, and there's maybe one that's 80% of the signal, and then so it's like having multiple jigsaw puzzles that spewed out together. Yeah.
Over here.
Your bacteria have been here since the beginning, and I wonder whether you have any sense at the moment of whether they're just being opportunistic when they invade like mammalian bodies, or are we dependent on them? Is it an essential commensal relationship?
All right, so the question is, bacteria have been around for a long time, and when they invade, are they just being opportunistic, or is it more of a symbiotic kind of relationship? Is that?
I think that was it. Yeah, both. I think is true. So bacteria have been around a lot longer than any higher organism. Like sharks don't have toll receptor 4, right? So sharks are just loaded with these bacteria that kill us. Does nothing to the shark. But it's also true that we rely, and you see this in mainstream medicine, my wife works for a naturopath, so naturopaths are more likely to believe, that there's this gut brain axis or gut hormonal axis. Axis is in AXIS, in case you didn't get my accent. But the scientific literature is full of anecdotal studies where, people who had cancer had different microflora in their gut, or, people who had any number of different maladies had changes to their gut microflora. And Jen and I have a colleague, Lisa Reynolds, who's an expert in microbiomes. We should get Lisa up here at some point, if somebody's listening. So, yes, I mean, it's true that the bacteria are just being opportunistic, like the Yersinia pestis, right? The bacteria were happy in a cold environment, and then the planet warmed up, and they became dangerous because they're just remodeling their membranes to survive, right? But in mammals, it's deadly.
Another question? Yep. Back at the bar.
Could you explain the use of PCR and the fact that it was never designed to diagnose anything? What is it actually used for? It doesn't diagnose disease.
Yeah, so the question is, PCR, what is it used for diagnostically currently, and why?
Yeah.
About,
I know what it's used for, but I don't know it wasn't designed for that.
right, okay,
Yeah.
yeah.
Yeah. So with PCR, it's basically a genomic assay, right? So if you don't know, you basically ask the question: do I see a certain set of bases that are unique to a particular organism, for example? And so the reason it's not more prevalent in hospital labs is, well, there's cost. Like the protein mass spec assay that I described, you don't need to know ahead of time what you're going to look for. With PCR, you need to design what's called a primer, and then that primer is the opposite of the thing you're looking for, and it pulls it out of solution, right? So it's more expensive, it's more sensitive, but it also generates more false positives. And so while it's quite amazing, really, that we can do that, it's not made it into mainstream diagnostics. It's used in certain cases where there is no other way to detect the presence of, say, an organism, and you just have to live with the fact that the false positive rate is higher. But yeah, I think the cost and the false positive rate are reasons why it's not gone into sort of mainstream clinical diagnostic labs.
Over here?
You were talking about bacteria changing their lipids depending on circumstances and so on. Are they actually making different lipids, or is this just change of isomers? Something along those lines.
You can repeat it. You probably understand it more.
Jen doesn't even want to try to repeat your question. So the question is, he used isomer, which is a chemical term. So the question is, are the bacteria, when they remodel their lipid membranes, are they making completely new lipids, or are they just making isomers? And the answer is both. So an isomer might be an example where instead of my ring finger being here, it's now over here where my pointer finger is, right? So the whole thing still weighs the same, but the configuration in space is different, right? So they do that, but they also might lengthen one of the fatty acids to make it too long to actually sit in the cleft. Or because my pinky and my thumb are phosphates, remember, they might make this less electronegative. So it might become more electropositive, and then it actually doesn't sit in the cleft properly. And there's actually an interesting story that I completely forgot to tell, which is that these same lipids are used as vaccine adjuvants already. And what happens there is they get rid of one of the phosphates. And so now this molecule that's designed, it looks like lipid A, it's just got one less phosphate. It doesn't sit in the cleft properly. So it wiggles around too much. but it's enough to send a signal to your immune system that something's going on, and so it helps recruit immune cells to the site.
At the bar?
I'm curious, I remember a couple years ago I read a story about how artificial intelligence was able to comb through medical research papers and do a pretty good job of diagnosing some things. I'm wondering, especially since a few weeks ago we've heard about quantum computing and artificial intelligence, how is it directly impacting your work or your colleagues?
Okay. Yeah. So the question is, there's people online, you said?
There are people online.
Wow. Hello, people online. So the question is, how is artificial intelligence? Quantum computing is just a way to speed things up. So I did worse in physics than I did in chemistry, so I won't address that part of it. But artificial intelligence is, in order to get money to do research, we use publicly funded research money, right? So this is why it's important for us, I think, people like me to try to translate what we're doing into something that's useful to society. And we just wrote a grant. Most of my research money comes from the NIH. You probably heard of Anthony Fauci because Trump was all over him when he was president. But Fauci was in charge of the National Institutes of Allergy and Infectious Disease. And so most of my research money comes from what was Anthony Fauci's institute at NIH. And so we just wrote a grant where one section of the grant is to use artificial intelligence to try to help us understand structures better. But in order to do that, we need to move from a few hundred structures to a few thousand structures. And we think we can do that. The grant is about doing things faster. And we have a completely different assay that I didn't talk about that will let us generate thousands of hypothetical structures, right? And then once we've got a much bigger library, then things like machine learning is another technology that's related to artificial intelligence. But it's basically like the question you were asking about, how do we get it right? Well, we need a library of sort of gold standards where we're pretty sure we got it right, and then we can teach the computer, and then the computer can teach itself from things that it's never seen before. So yeah, we are using it. It's just not mainstream at the moment with what we're doing, because it's a little bit too fundamental, and we just don't have enough data. You really need giant piles of data.
Any more questions? I think we have time for one more, if there is. Yep, they're at the bar.
Yeah, I have one real quick. Cell membranes. So my understanding is that cell membranes are selective about what gets through, and a lot of therapeutics are trying to piggyback chemicals onto things that'll knock at the door of the cell membrane and get through. So if that's the case, and maybe that's not right, have bacteria just evolved to get through the cell membrane? Because isn't that the first line of the wall to protect yourself? I don't know.
Yeah.
I'm-
Are you going to
Yeah, so the
Come on, help me out here.
so the question was, isn't the cell wall the line of defense against bacteria? Is that correct?
Well, yeah, against anything coming in. And how does
Yeah.
therapeutics tie in with that? Because I know there's a lot of work on how basically to get through the membrane wall to deal with the cellular level.
Yeah. Okay. Sorry, people online. Yeah. So, I mean, bacteria, like macrophage, is a cell that is like Pac-Man. They're in your lungs. It'll just gobble up bacteria and destroy them, right? Unless there's too many, and then they get overloaded, like I was telling you. So there's that mechanism to clear bacteria. They basically just get destroyed, broken down into components. And in terms of drugs to kill bacteria, it's a complicated question. That's why I'm having trouble answering it. But it's a good question. Bacteria, there have been attempts to, like the pneumonia vaccine, right? So the pneumonia vaccine works because pneumococcal bacteria are stable enough that we can pull out antigens. Now the vaccine's got 20 different strains, right? So we can pull out antigens from each of those and basically inoculate people, and you make your own antibodies to kill those bacteria, right? They bind to the antibody and then they get cleared through a mechanism. The other way, but the problem in many cases is the bacteria mutate too easily, right? And so you can't just pick one surface protein / antigen. An antigen is just anything that you can make an antibody against. So you can't just pick one if the bacteria mutates a lot. So a friend of mine, a Canadian who now sadly is deceased, but spent most of his life in the States, he was trying to develop antibodies that would kill Staph aureus. So you've heard of methicillin-resistant Staph aureus or MRSA. So Mark Shurtleff had developed antibodies against 6 different surface proteins to staph, and he could not kill it, right? So he could knock down like 99% of it, but he couldn't create what was called sterilizing immunity, which means you've killed all of it. And so the 1% that's still there, has mutated and grows again. So the problem is they are the ultimate survivors because they have been around so long, longer than we've been here. So they use mutation to their advantage. There's even there's a organism called Radiococcans durans. I may be mutilating that, but it's found in the nuclear waste tanks at the Hanford site in Richland, Washington. And it's got like 12 copies of its genome, and it just keeps getting irradiated, but it's got so many copies that it can keep up and constantly rebuild itself and it basically survives in this really harsh climate. So yeah, I think that they just mutate quite easily. This was the problem with COVID. It mutated. It's a virus, but the surface is where the antigens are that your immune system recognizes, and it just mutated quite rapidly. Okay, I mutilated that answer. I'm sorry.
Okay, I'm going to turn the mic over to the Dean of Science, Peter.
Uh-oh, it's my boss.
Well, thanks, Dave. I'm going to stay away from the microphone now that I know that you breathed on it. Also, I don't think I'm ever going to take a shower again. So Dave closed his talk on talking about big money. I represent small money. I'm the Dean of the Faculty of Science. I want to point out that this event is free, and it will stay free, just to be clear. But it does cost us about $5,000 a year. And we don't have a base budget for that. The university is not funding the Faculty of Science to launch these events, sadly. So that is, we are taking the money from other places, funding graduate students, funding undergraduates, and so on. And I'm saying all of this, and you know which way this is going. I know you do. So if any one of you feels particularly generous, or you have a rich friend, talk to me. Please do. We would like to build enough funding to maintain this basically the series indefinitely. And it's not that we're paying our speakers here, right? They're doing this because they're kind people and smart. So again, if you want to talk to me, you can send me an email. I'm going to hang around for another 5 or 10 minutes or so. I'm very easy to Google. Dean of Science, University of Victoria, right? Yeah, Dean of Science is important because there are 11 of us, and I don't want the money to go elsewhere. Thank you.